Okay. So um, I don't have your bio up in front of me, Will, but I sent it out with a notice. Will is a studies care. I'm sorry, I'm having a blank.
Not spiders.
Not spiders, carabids. And you're a California person, so you're at least not having the time zone problem that some of our speakers have. We've had speakers from Australia and other parts of the world had time zone issues. So you're going to be talking about carabids. So I will just turn it over to Will.
Great. So let's get the screen shared here. I think I can do that. All right. Does that look okay to you guys? You got to see the full screen? Okay. And I need just to rearrange things a little bit so I can maneuver. Okay, well, thanks, Blaine, and thanks for the invitation, and it's going to be fun to talk to you guys. It's I've been working with ground beetles for, probably close to 30 years now. And so this is something that I'm, really passionate about. I love to talk to people about it. And I thought that with this group, rather than sort of focus on a single research question and kind of go through kind of the usual presentation that I might for some venues, that I would just sort of give some overviews and dip into some little bits of the various research that I participated in and some of the fun results, and talk about some of our local carabids of interest that you guys might be able to find, and some of their habits and things like that. So it's kind of structured in a way that, like, if there are questions that come up, I'm not great at monitoring the chat, but somebody can get my attention and we can definitely segue and talk about things. So that's why I picked this title, sort of ground beetle vignettes, but it's more than just ground beetles. And so the initial part here, you guys, many of you this will just be a review, but it's always good in case you have lepidopterists and spider people in the audience to remind them what beetles are and which ones I'm talking about. So, among the huge array of beetles, we divide them into four suborders. These are two are small ones. So Archostomata, these are fun things that you can collect up in the Sierras. There's just a few of them. There's a great fossil record for them, but not many species. Mixophagens, which are these tiny little beetles, typically, that are odd. It's a very, very small suborder. And then the vast majority of species are in Polyphaga. So that's most of the beetles people tend to see. The second largest suborder is Geadephaga, and that's where the carabids are. So this is a group of beetles that has been recognized as a group, a natural group, almost the same as what it is now, all the way back since Latreille, first set up categories of beetles back in the 1800s. So they're very recognizable. They're found throughout the world. There's about 40,000 species worldwide in Geadephaga. And we think of them as divided up into Hydrogeadephaga and Geadephaga. So what I'm going to talk about is the Geadephaga, but this gives you an idea of how they're all sort of nested in the whole group here. It's the Geadephaga, and then Hydrogeadephaga are these water beetles. That's a few thousand species out of the pile. And so I'm not going to talk about these guys. It's not a natural group. They're a paraphyletic group. And then the Geadephaga is everybody else, which is the vast majority of the species. This is a natural group, and it includes the families of tiger beetles, Cicindelidae, the Trachypachydae, and the Carabidae. Recent classifications probably still tend to have the tiger beetles nested within the carabids, because that was a lot of the evidence that we've had supported that position. but I know of some genomic stuff that's coming out that's going to, put them back out in their own family. So I've started treating them as their own family, and they're a very distinctive group. So you might see that and say, oh, aren't they actually carabids? And the answer is yes, sort of, until those papers get published, because that's kind of how we follow the classifications. So what I'm going to do is dip in a little bit on each of these with a story or two, and then more stories about the main carabids. And I'm just going to work my way up the tree. So the Trachypachydae and the Cicindelidae are these groups that lie outside of the carabids, which include a whole bunch of mid-grade groups. So I've just sort of reduced this to a few branches so you can see them. And then the vast majority of species are in this one subfamily, the Harpalinae, which is 25,000 species out of the whole big pile. So let's dip in a little bit here. So the Trachypachydae, it's a really fun family because you can actually go out and collect almost every specimen or every species in the family, which you can't do that for a lot of beetle families. In Trachypacus, this genus, there's only 4 species, 3 of which are in Western North America, and one really rare one, that'll be the hard one to collect in Europe. But 2 of these are pretty commonly collected in California if you know where to look for them. And then the other genus in the family is Cystillosoma, these beautiful things that are from Chile and part of Argentina. But there's only 2 species in them. And this is a really old family. There's a good fossil record of these guys, but the overall diversity is really diminished now. Now, we've done some research work on these guys and their defensive chemistry and stuff, but that's not as exciting, I think, as going out and actually collecting these guys. So I thought I'd mention a few strategies in case you're interested in finding them. Trachypacus gibbsi is one that you can find pretty commonly up in the Sierras. If you get up into the higher elevation, say Sonora Pass, especially, now this is a little later in the season, but you know that time of year when you go up in the Sierras and you look in the shade and you can still see snow out up underneath the trees. And sometimes you'll find a little bit of snow in a donut around a tree, and it'll have a bare area that when the sun has warmed up the tree and it's warmed up the soil, around that you'll have this really loose, fluffy duff that's kind of moist. And if you come out during the day and you just watch for a little bit, you'll see little shiny beetles kind of jumping in and out of this duff. And so Trachypachyds have this thing where they sort of swim through the soil, and they have really kind of odd legs the way they work. And you can see them doing that, and you can collect lots of them at the right time of year. As it dries out, then they tend to disappear. Trachypacus inermis then has a big range. In fact, it goes all the way south to the San Jacintos and way up into Canada. And you can find it in a variety of places, but also that loose duff again. So these guys really are kind of like soil swimmers as well. This is in Oregon, just along a road cut. You find a road cut, you get there in the morning just as the sun is warming it up. And again, you start to see these little metallic beetles sort of swimming in and out of the soil. If you do want to go get the 3rd species that we have here in Western North America, Trachypacus sleveni, you just need to go to Oregon. This is Cape Perpetua up along the Oregon coast. And again, see, this is a like exfoliating dirt and rock along there. So it's like that loose material, and they just sort of are swimming through it. Even you go to Chile to the Cystillosoma sites, same sort of thing. So this is near Villarica in Chile, same sort of thing, different genus, but the same sort of behavior. So they can be collected other places, but this is one of the best ways to find this kind of really obscure little family. Okay, so I'm not going to say more about the Trachypachyds. I'm going to just say one little story about tiger beetles and not spending time really introducing them, because pretty much everybody is familiar with tiger beetles, right? They're day active, they're beautiful things, lots of collectors. Like For carabidologists, we kind of think of them as like the butterflies of our family, something that we can chase during the day because most of our beetles are nocturnal. And this is something we published recently, which I think you might find interesting. So here's a megacephaline tiger beetle, Tetraca carolina. You can get them in the very southeastern part of California, and much more commonly as you go east. So if you get over into Arizona, they're not so rare. It's quite a attractive beetle. And then on the other side, there's this strange looking object. And this strange object is actually a piece of amber with a beetle inclusion in it that was shaped like a vase and apparently used as a piece of jewelry. And originally, Walter Horn had seen this piece of amber and described the beetle that was inside as Tetraca carolina, suggesting that this is the same species that had been around for more than 30 million years, if it in fact was, encased in amber. And this is Baltic amber, so that's 30 to 50 million years old. This was described. A few people looked at it, and a bunch of people commented. Some said, oh, is it really that, or is not that? And then it disappeared. The fossil seems to have disappeared. We don't actually know where it got to, but then just a couple of years ago, it was returned to the museum in Berlin, kind of no questions asked kind of thing. It just sort of appeared. But now the inclusion is highly occluded and we can't really see it. But it's an important fossil. And so my colleague, Joe Schmidt in Rostock, got a hold of it so that we could study it to see, is this really Tetraca carolina? Like, can a species of beetle actually endure for 40 million years and not change? We certainly had doubted it, but we couldn't see a lot. So with our colleagues there at Rostock, we used the CT scanning technology, and they were able to peer through all of that occlusion and get beautiful pictures of the beetle that's inside. And additionally, then they were able to construct these 3D images, which you can manipulate and actually see all of the details of the fossil that's inside. And we shared these with the publication so that everybody can see exactly what kind of information we can see when we were studying the fossil. So as it turns out, it was not Tetraca carolina. It's actually Paleoiracina. It was another fossil species that Joe and I had described earlier. We kind of suspected that it might be because the age was right. So we had this chance to actually get the fossil back and then use this really fun technology to be able to see all of the details that we couldn't have seen otherwise. So that's a couple little vignettes about some of the, I think of outgroups of the carabids, which is really the group that I'm really interested in. So, tiger beetles and trachypackets are out here. And then there's these early branching groups, some of which you guys are probably familiar with, things like Omicron, the round sand beetle that doesn't look like most other carabids, Carabus, which is another popular group for collectors to collect, especially in the old world. I'm going to talk a little bit more about some of these, like Promechognathus, which is one of our Californian species, and Brachinus, and then I'll jump into some of the harpalines, which is really the groups that I work on for research most of the time. So the early branching groups, what I want to do is talk a little bit about some of the chemical ecology and chemical biosynthesis work that we've done, because it's really fun. Like chemical ecology, aside from systematics, which I love, is the next best thing. Partly because you can just have fun in nature with it, right? Especially with like defensive chemistry. I can find a bunch of Formica ants somewhere, find one of my carabids, agitate it so that it actually shoots some of its defensive compounds out, drop it in the middle of the ants and watch them all run. So you can actually see chemical ecology in action. It's kind of fun to have the chance to do that. For those of you that haven't seen what's going on, all of the Geadephagan beetles have defensive glands, defensive pituitary glands. So this is a view of one of our Californian species. This is Platinus brunia-marginatus, a very common beetle in any sort of little riparian areas throughout the state. And if you pick them up, they smell, quite acidic and sharp. And they're producing formic acid. So the formic acid in these reservoirs, which is this sort of partly translucent structures you see in the abdomen, can be about 60 to 80% formic acid. So it's really very potent. And it takes up a huge amount of their abdomen. So they put a lot of energy into making this formic acid and storing it for defense. So all Geadephagan beetles have that. Now, one very special Geadephagan beetle that probably most of you are somewhat familiar with are bombardier beetles. And so I'm going to try and show just a little bit of a video that KQED did in my lab to kind of give you an idea of bombardier beetles in action and sort of close up in case you haven't seen it. Okay, so let me know if this isn't working for you. Is it okay?
We don't
No.
see the video.
You don't see the, okay, so
You
why?
you have to do new share probably.
Okay. I will. And sound is on. Can you see the video now?
Yes,
Okay. And let me know if you can hear it.
we can hear it.
Okay.
The bombardier beetle is one of nature's most improbable creatures. For some, it throws a monkey wrench into the whole idea of evolution. This beetle is a walking powder keg. That cloud you see is the result of a lightning-fast chemical reaction inside his body. When attacked, the beetle sets off the explosion, releasing a boiling hot, stinging poison that sends his enemies running. He can even aim it. This potent
So there's several minutes more, but and if you haven't seen, you should check it out because the deep look videos are so great, and they have more footage. But I want to definitely get to the point where it was able to repel that spider, because I thought you'd appreciate that. So let me redo my share now, and this is in my way. Okay, are we good again? We can see the full screen?
Yeah, it's good.
Yes.
Yeah, okay, perfect. Okay.
Actually, it's
So,
not really full screen, but it's good enough.
oh, it's not full screen?
Now we can see your
Oh,
your
I see.
controls too.
It's sharing the wrong screen. That's why. There we go. How's that?
Yeah, that's
yes.
great.
Yeah. Okay, that's all righty. Okay, now we're cooking. Okay, so the bombardier beetles, right? But I'm sure many of you are familiar with. In California, we have two kinds of bombardier beetles. We have what you saw in the video, which is the genus Brachinus. We have many species of Brachinus, and they're common along larger streams and rivers. As larvae, they're ectoparasites of the water beetle's pupae. So that's why they hang out there. Although there's many species, they tend to look very similar like this, with this sort of orange forebodies and bluish black elytra. And then the other bombardiers that we have in the state is Metrius contractus. So there's just one species here. They're not closely related, although their chemistry is very similar. And these guys are common through the Sierras and much of the Coast Range. And you can find them in Oak Bay Forest pretty commonly during the season. The bombardiers This is the Brachynes version of the glands. So they have a set of secretory cells that produce the hydroquinones and hydrogen peroxide, and they fill those reservoirs. So just like the reservoirs that I showed in the other beetle a little while ago, they have reservoirs like that, but they're filled with hydroquinones and hydrogen peroxide. They also have these extra little structures here that are reaction chambers. So heavily sclerotized set of chambers that hold these enzymes, catalases and peroxidases. And so when they muscles squeeze the reservoir and it shoots a little bit of this hydroquinone hydrogen peroxide mixture in there, it picks up these enzymes, excuse me, and then as it's being ejected, this chemical reaction occurs. So you get quinones, water, oxygen, and lots of heat. And so they pulse this at a very high rate and can shoot 10, 12, sometimes as much as 20 times. So it's quite an arsenal that they can actually deploy in defense. So this was a fascinating system. I was a grad student when I was first introduced to this, and I knew that somehow or another I was going to have to work on it. So we had a project for a number of years where we really investigated a lot of the details of this system. And so one of the ways we do that is that I would do lots of dissection. So here's a bombardier beetle gland up here at the top, and I've ligated off the reservoir. So I just pull a hair out of my head, and I could then tie this off so that it didn't release into the reaction chamber, dissect it off so we could see exactly what the contents are unexploded. And then we can do fun things like capture the spray. So here's a beetle down here. I coax them very gently into a Pasteur pipette, and then I introduce a little bit of CO2. And if you watch carefully, you'll be able to see it spray. And then we can dump the beetle out and then wash that out with some solvent. And so we can get the before and after the contents and see how it changes. So the GCMS up here, we got hydroquinone and toluhydroquinone. So that's our hydroquinones. The reaction occurs, and then you get big peaks here at benzoquinone and toluquinone, right? So this is a way we could get a bunch of these guys into the lab and then manipulate them in various ways. And so one of the ways we manipulate them is that we would force them to spray. Then we can inject them or feed them precursors that are various kinds of isotopes. So a precursor that has deuterium instead of the hydrogen there that we have made or we made ourselves. So we knew exactly what its structure is. Give them a little time to cook and make some more juice. Then we would force them to spray or take their glands out, and then we could check it again. And in that process, we can see where they incorporate the precursor by looking at the difference in retention time between compounds that we gave them with the deuterium in it and the normal stuff. And in that way, we could know exactly how they put together the molecules when they synthesize the compounds. And then the chemists, I don't do the chemistry part here, but the chemist would then be able to work out the steps in the biosynthetic pathway. So we began to learn a lot about, not just their behavior, how they spray it, but like, how do they actually make this stuff? And so without, boring you with a whole bunch of chemistry, just to say that as science goes, like, you always get surprises. Like, it turns out there's actually multiple pathways, at least 2 pathways we discovered. So they have a much more complicated system than we actually originally thought. That the original hypothesis, they just make a single kind of quinone precursor or phenol precursor, and then they make all their different products from that, but they actually have multiple different pathways. The other kind of fun thing that we haven't published on yet that I started doing was feeding them things that don't occur in nature. It's kind of crazy, but these beetles are so good at detoxifying their food and taking most phenols that we would feed it that are really kind of nasty chemicals, that they can turn it into products that never occur in nature. So here's a beetle. They just eat dog food laced with stuff. So I laced it with 3-nitrophenol, which is not something they would ever encounter otherwise. And you can see they've actually produced their quinones with these nitrile groups on them. So they're bright yellow and they smell different. And so this gives us a little more clues about how they're synthesizing things, but it just became kind of a thing I was doing. It's like, well, will they eat this? Will they eat this? And it turns out they eat, and they can make all kinds of crazy things out of it. So bombardiers are a really fun part of the biosynthesis and chemical ecology research we've done. The other mid-grade group that's here in California that we've worked on is Promechognathus. And you'll see Metrius comes up in this story too. That was the other bombardier that I was talking about. But the research was really focused on Promechognathus, which are these really cool, sleek-looking beetles with big jaws. And they're millipede hunters. That's their specialty. So I had a student who worked with me. We did a bunch of behavioral and chemical ecology work with these guys. And so this is one of the 2 species of Promechognathus we have. There's Promechognathus levissimus and crassus, and they specialize on millipedes, particularly in the Bay Area. They're after the fluorescent millipedes in Zistiker. So you can go out with your UV light and say January through early April, especially during the February, March, when it's nice and wet out there, and the leaf litter will just light up with your UV light of these things, and you're gonna find Promechognathus hunting them out there. And they have this really cool behavior where they stalk them and they sort of mount the back of the millipede and ride them, and they begin to bite into them. What we found is that there was an earlier hypothesis that they would grab them and bite them quick enough so that the cyanide was not released. So these are cyanide protected millipedes. And so the idea was that the millipede hunter would then stop the millipede from spraying to avoid getting it on them. But we did a number of tests and we realized, no, that's not the case, that like these millipedes are just spraying like crazy and the beetles don't care. So we'd say, well, there's something going on here, because most other insects, they really don't like the level of cyanide that these millipedes can produce. So how is it that they're going about not being toxic, get the toxic effect of the cyanide? So we did a lot of behavioral things to determine that it didn't seem like they were doing anything to avoid it. So we said, well, I guess they're just tolerant of it. And so then we set up these experiments, which are our cyanide test chamber, or as we, refer to it in the lab, the death chamber. But it actually wasn't for Promechognathus. We couldn't kill them with cyanide. We tested it on a bunch of other beetles. It actually, you could knock them down and get them out. You didn't have to kill them, but you could see a response for most things within minutes. So here's a little bit of a complicated chart. We looked at a bunch of different species that are things that occur in the same habitat with Promechognathus and the millipedes. We also did some feeding trials to see if they would eat the millipedes. The majority of them won't even touch the millipedes at all. And then we got a variety of things to look at body sizes, because Promechognathus is relatively large and you would expect more tolerance as they get bigger. That would be normal, and that is truly the case. These are arranged more or less by body size, up to some quite large beetles, much larger than the size of Promechognathus. And this is the amount of cyanide we introduced into the chamber. And so we got to the point that we just couldn't add enough to do anything. Like they just didn't care. Like they weren't happy they were in a chamber, in a little cup. But unlike these other groups, you can see they're just knocked down really, really fast. And even the big ones eventually get knocked down, except for Metrius contractus, which was, again, this is another one of those cases we were totally surprised. We had no expectation that that would be the case. And in fact, we still can't explain exactly why. There's no indication that Metrius feeds on the millipedes. We've never seen it happen. They would kind of eat a little bit of dead millipedes in the lab, but they don't seem interested in live millipedes. So we don't know why, but they also seem to have innate resistance or tolerance to cyanide. Here's both species of Promachognathus. They're entirely resistant to it. So a fun little group, Promachognathus here in California, that turns out to have this really interesting chemical ecology story. Okay, so these are some vignettes from the mid-grade. Now the Braconine bombardier beetles here, they're the sister group to the huge clade in our phylogeny of all of the derived carabids in the Harpalinae. Right? So there's 25,000 or so described species. That's probably not more than a third of all the species that are to be described in the world. And this is the group that I my main interest is for systematics and taxonomy. And I work in the pterostachines, which are a species-rich group, which makes up about 15% of the entire family. And that's kind of in its broadest sense. And so as it turns out, the pterostachines, as people know them, not surprisingly, once we started really looking them, turn out to be many groups scattered across the subfamily Harpalinae. So there's a bunch of names here. Don't worry so much about that. These are all tribes within the Harpalinae. And then where I've put the red arrows, those are all the places that, since I've been working on it, pterostachini now comes out. So it's this huge set of groups, but they're actually not each other's most close relatives, right? They're not a monophyletic group. The chunk I will talk about that are in California, though, do form a monophyletic group. So they're here in this greeny brown representation of the clade. And this is all the stuff that we would find here in California. So I'm going to introduce some of these taxa to you, so you might find them interesting. I might have a chance to see them. So one that you're certainly going to see, and many of you probably have already, is this tribe, the Zabrini, the Zabrines. And Zabrines are these guys, you've seen them probably just out running on the sidewalk. Some of them are common enough. Some are introduced species that you can find them in parks. You can find them in lawns. So this is the genus Amara. And they definitely have a look. Most Amara and close relatives have this similar sort of look to them and are common throughout California. Even in the quite dry open habitats in California, we have native Amara species that can be really abundant. One of the things that they do that's somewhat unusual, it's not common in carabid beetles, is they are seed and pollen feeders. And so occasionally this is an Amara. You can see it, climbing up a grass stalk to feed on the grass seeds and pollen. So while most carabids are predators and scavengers, the scavengers will eat an awful lot of different things. Zabrines are not necessarily exclusively pollen and seed feeders, but largely pollen and seed feeders. Probably can't produce very many eggs if they don't get that in their diet, but they might also eat a few other insects if they don't have to chase them down. So that's the Zabrine, something that you'll see even around your parks and stuff in town. So all of the pterostachines that most of you that are familiar with them know of are going to be in this group here, the Pterostachina, the subfamily of Pterostachini, kind of in its narrow sense. And this is a clade that includes all the northern hemisphere taxa that are related. And A few of these are centered in North America and just trickle across the equator in the edges of their distribution. Whereas this group, this is the Euchroina, this is a decidedly southern hemisphere group, so South America and Africa, Australia. So they're sister to this northern hemisphere group that includes the pterostikines that most of us are familiar with. So this is the genus Poecilus and Pterosticus that I'll talk about a bit more. So that's what I want to do. I'm going to go through some of our West Coast, our Western North American Pterosticus and pterostikines that you guys might see that you might find interesting, with a little information about their natural history and their distribution. Just to, when you're out and about, you get a chance to see them, that you might find it kind of fun to check them out. So this is an odd Pterosticus that's quite rare. I'm sorry, it's not a Pterosticus, but a pterostikine. It's in the genus Stomus. And so here's a distribution map. So here's Crescent City, way up in the northern part of California. So, up around Del Norte County, and right along the coast into Oregon, up to just a little bit south of the level of Eugene. This is the range that Stomus termitiformis is known from. And they're quite rarely collected. And they're found in these nice, lush myrtle forests up along the coast. And probably in some of the lusher mixed forest in the northern part of California. And what makes them, there's a number of things. They're kind of just cool looking pterostikines with big heads and kind of a very graceful looking body form. But this is the only species of the genus in North America. The only native species. There's an introduced one, Stomus fumicatus, that was introduced from Europe. And then there's a bunch of Asian species. So this is our only native Stomus. And so I pursued this for quite a long time before I finally got some specimens here where I put the little gold star, and was able to study it in detail and sequence it to sort of verify that this is in fact true Stomus, and it really does come out together with the other Stomus, with a very peculiar, maybe relictual sort of distribution. This is a beautiful animal that you can get up again in the boundary between Oregon and Washington. This is Pterosticus johnsoni, and the image really doesn't do it justice. Like, it's a beautiful, brilliant metallic purple on the elytra, as these huge foveae. And for a long time, people referred to it as the waterfall beetle because it was found near waterfalls, but it's not exclusively found near waterfalls at all, but fast rushing cold water, just right along the rocks near the water, which is a very unusual Pterosticus habitat. To find beetles this big. So it's a big beetle and it's quite stunning. And you can find it really with its feet wet right in the stream bed. A little bit like that, another northern group that just trickles into California is the Pterosticus in the subgenus Cryobius. So they are also riparian, but not like Orso johnsonus, which are literally like standing right in the water. These guys would be in the wet, soggy bits. But even, I think, kind of more interesting is that they go all the way up into Alaska, and they get much more common up in Washington and Idaho. And you can just find them walking on the edges of snowfields and glaciers. They just don't do warm weather. They like it where it's very cold. And so they have the well-fitted name Cryobius, where they love to hang out on these really cold habitats. So sticking with the sort of odd riparian habit of Pterosticus, this is another one, Pseudopheronina. And so this is me. I'm up here, this little silhouette you can see, and this is like a near vertical it's not a proper waterfall, but it's like a trickle of water that's pretty constant. And this is the only place where we find these. You have to climb up these extensive erosional seeps and then dig around in the dirt and are in the wet soil around the edges of it, together with their larvae living in this rather peculiar habitat. And they're found in Oregon, in this sort of situation. So if you get up to Oregon and you're near Corvallis, you can go out to Mary's Peak. It's a fantastic place to collect out there. Lots of carabids, but lots of other groups too, in this very lush sort of habitat. Okay, that's all looking mighty lush and green for us Californians. Let's look at some of the California taxa that we have here, where we endure this Mediterranean climate where we have extensive periods of dry, and our taxa are really adapted to that. So in California, the vast majority of pterostachines that you're going to pick up are going to be in one of these two subgenera of Pterosticus, Leptophonia and Hyperpes. Right? There are a couple other genera and subgenera here, but they're much, much less common. The vast majority are going to be these guys. Of the pterostachine looking things that you might find, you can quite easily, even with a hand lens here in California, pick one up, take a look, and if it doesn't have a seta on the disc of the elytra, then you've got Leptophonia or Hyperpes. So if you check any of the other groups, what you'll see is there'll be punctures, a few, at least one, up to 3 or 4 punctures that have a little hair on them on the elytra. And so you can know right away whether you've got one of these guys. And these are pretty classic beetles. My guess is any of you that have been out very much looking around, you have already seen them, whether you are familiar with exactly what they are or not, but they can be incredibly abundant. One of the most important arthropod predators in the soil and leaf litter in California. So this is a thing called Leptophonia inanus, which is one of the most common ones in much of the Sierras. It goes all the way from Kern County all the way up into the Cascades. And they love this woody debris that is so common up in the Sierras. And so if you go up just after the snow is melted, when this woody debris is nice and wet and you start rolling it around, you're almost certain to run into Leptophonia inanus, which has this nice cylindrical form, and it's not flipped over. If you turn it over and if you look at the hind legs, you'll see it has this, in the males, it has this ridiculously long pointy trochanter sticking out, which is a really strange leg structure, but it's found throughout its range to have variation in the length of this, but always really, really long. In our oak woodland habitat, we have other Leptophonia. This is sort of a classic Bay Area oak woodland habitat. This is a Leptophonia caligans, which is, as Leptophonia go, a fairly large one. The Leptophonia tend to be small and brown, with the Hyperpes are big and black. There's some overlap, but this is a relatively large Leptophonia with kind of a big head and tiny little eyes. And so one of the things we found in Leptophonia is that eye reduction and more of a deeper soil habit has evolved at least 4 times. And so in the Mediterranean type climate that we live in, with these long dry periods, it seems that these have evolved to sort of, get down to where the moisture is and spend less and less time coming up on the surface. So what do you need an eye for? And if you look carefully, you can see there's just a tiny little dot here that's the eye. This is inanus, so a proper sized eye. This is almost gone, a fairly small one, nothing but a little tiny dot left there. So caligans is here. So this is one of the times that we see it evolve, and there's a couple more times that we see this general form of being kind of cylindrical, tiny eye, and being subterranean, and only coming up really in the wintertime, or occasionally people find them popping out in caves. They don't seem to live in caves, but occasionally that's where people collect them. So then there's Hyferpes, right? So that's an introduction to some of the Leptophonia. Hyferpes is really the dominant Pterosticus in California. And so this is a generalized map of the distribution of all the Hyferpes species. So the vast majority are in the West. There's just 2 species in the Eastern United States. And these numbers kind of roughly center on the area where the diversity is about this number. So you can see California has, 75 species within its border. About 60 of those are endemic to the state here. It's probably at least 24 or 36 undescribed species yet to find here. So it's really a hot spot for Hyferpes. And as you move away from it, then from California, then the diversity diminishes quite rapidly. So, into Oregon and Washington up the coast, you could still have 20 or so species. Then it kind of peters out, one reaching Alaska. The Great Basin, there's just one widespread one, a few in the Rockies, and then they trickle down into the southern end of the Rockies into northern New Mexico and Colorado. But really, we're at the epicenter of Hyferpes diversity. And they really pack it in here in California. So here's from a single location up in the Sierras, not very far from South Lake Tahoe, where you can find 8 species all packed in an area not even 100 meters square. They all just living together and in abundant numbers. The smallest 2 out here are Leptophonia, and then all of the rest of these are Hyferpes, with the big Hyferpes out on the end, which if you know any Hyferpes, you probably know this monster. This is the largest Pterosticus in North America. This is Pterosticus llama. And then there's Morionites, and there's Serapes, and so forth down. And so it looks like in these areas, there can pack so many species of the same subgenus, really very similar, and they're parsing the niche by size, right? So there's a size range distribution that really is largely non-overlapping. If we go to oak woodland habitat, we find fewer. I think that the nutrients and stuff in the Sierras are just so much more abundant. Like 8 species can coexist there. We see 4 to 6 species is typical in, say, the oak woodlands of the Coast Range and the Diablo Range. This is from the Diablo Range. We also see that the size strategy doesn't hold up so much. There's Californicus, Vicinus, and Protensiformis, and they broadly overlap in size. Now, Protensiformis tends to be the bigger one, Californicus kind of the medium, Vicinus the small one, but the overlap is really quite extensive, and a little Leptophonia that's quite a bit smaller. We did a lot of studies. This is from a 5-year pitfall trap study that we did out in Mount Diablo. There is quite a bit of overlap in their activity period, although they tend to be slightly different, with Californicus being generally out more frequently and being more bimodal in its distribution, and Vicinus being a little bit more narrow, and Protensiformis even more narrow. If you look at the habitat structure that we find them in, Californicus is kind of everywhere, but primarily in quite open habitat. Vicinus everywhere, but mostly in the light tree cover, and Protensiformis is in the light tree cover and the deep leaf litter, and then Angustus is really just winter active in leaf litter and doesn't seem to overlap with them. So a little different strategy, doesn't seem to be as simply explained as just body size with these guys. So something else that turned up that really surprised us during this study is that, having looked at these guys for decades, and I suddenly started to realize that there's a funny little mark that I kept seeing on them that was very consistent. And as it turns out, it's not just a random scuff on a specimen in the pin collection, but these are actually very consistent marks exactly in the same places on the female elytra and the male last ventrite. And they're very consistent and they're very species specific. And so what it turns out is that it's some sort of behavior during copulation. The males are actually abrading the female elytra and wearing through the cuticle. And so, and it's species specific in terms of its position. And if you look at the males, there's a corresponding spot. The males mount dorsally, and this is where the ventrite interacts with the elytra. The length of the ediogus of them is different, so the point that they rub actually is different. And so this seems to be showing us mated and unmated individuals. The exact behavior, we still don't know. We can't get them to perform for us in the lab. They seem to be shy. But either there is an extended period where a male holds a female, perhaps mate guarding, and is vigorously rubbing to maintain that mating, or the females mate multiple times, many times, and because this is not a trivial like scrape. Like this is quite deep. It's through the outer layers of the cuticle. So currently we have another project going where we're doing dissections to look at breeding condition, looking at the eggs and sperm and stuff in these to try to get a better handle on exactly what's going on. So lots of hyperpes have sexually dimorphic pairs. So this started, really got us started looking at the males and females in great detail in hyperpes. A lot of them are sexually dimorphic, with males having lots of secondary sexual characteristics. So this is Pterosticus serapes, one of those Sierran species. This is the tip of the male abdomen, and you can see it's got this great big keel-like structure on it. And if you look at his hind legs, this is the trochanter, this is the femur. He has this great big, like tooth-like tubercle that sticks out. And the females have neither of these. So we don't know a lot about what the actual behaviors are, but we're beginning to realize that, lots of people look at it and goes, oh gosh, another Pterosticus. They all look the same. Actually, they are really have a lot going on. And so a lot of it has to do with these patterns of breeding that we're just beginning to understand. Size dimorphism also we've been looking at. So here's our friend Pterosticus llama, just a casual observation. We said, gee, the Pterosticus llama males always look bigger. So we did a bit of morphometrics to look at the size. And so this compares the pronotum and elytral length combined. That's our surrogate for overall body size on these guys, with males on the top in red here and females on the bottom in blue. We decided to look at kind of populations. I'll put that in quotes. We just had some arbitrary boundaries of what are kind of typical bioregions to see if there was any significant difference. There's a little bit of difference that in the Sierras, they're just generally larger, right? But this sort of pairing of big males and small females holds very consistently across all of Pterosticus llama. And then we looked at two other large species that are almost the size of Pterosticus llama, Amenetrizii and Morionides, and we see no such sexual dimorphism in size. So typically when a species has larger males, and that's the dimorphism, there means that there's some kind of male-male combat going on to compete for females, or some kind of female choice for large males for some reason. So again, we don't know the behavior, but boy, that's a very clear pattern of something going on during the mating behavior in Pterosticus llama. So a little bit about some more habitat. So I mentioned Amenetrizii as one of the ones we measured. It's a large one. This is unusual in terms of most hyperpes. We think of, forest and woodland species. These guys are not right out on the beach, but they're like sub-beach species that you can find in the back dunes and stuff like that. And you never find them in a forested area. It used to be quite common in the San Francisco coastal area. Those populations are gone, as you can imagine, too much disturbance. We have found some populations up in Inverness along the coast. They're still there, and there might be some populations farther south. We're not sure whether there's another species as you get as far south as, say, Santa Barbara, because there's a very similar form called Pterosticus glycicans that's out on the Channel Islands. So San Clemente Island, in these areas that are completely treeless, you find something that looks quite similar to this, but slightly different. It does seem to be a distinct species out on the islands there, and it is by far the most abundant and very abundant large Pterosticus on the island. There are also probably a third species in this complex. It's on Santa Cruz Island, again, in this treeless erosional habitat that you find there. And as soon as you go into the tree line on Santa Cruz, that species drops out, and you get another small leaf litter dweller called a Pterosticus inermis, or I put question mark, and because I actually think now that there's 3 things under the name inermis. And so in the Los Angeles area, you're going to see a species that's going to look very similar to this. It's a relatively small hyperpes with kind of a globose looking pronotum here. That's probably the true inermis. You can get them in the hills just northeast of town. But the male genitalia and the sequence data looks different out on Santa Cruz. And if you get up as far as Santa Barbara, you get something that looks like inermis, but then also has differences in the male and in the sequence data. So as we keep studying these things, more and more new species keep emerging from it. Okay, so I said a lot about that. I'd thrilled to answer questions. There's like 100 more species I could go through, but I think you guys have probably had enough. But I'd love to talk about any part of this stuff or answer questions about carabids if you have any for me. So I will stop sharing unless you need to see a slide, and then we can talk a little bit. So thanks again for inviting me.
Well, thank you very much for participating.
Yeah, this was great. I even liked all the taxonomy stuff in the beginning. That was really good, helpful for me.
Will, on the
I have a
bombardier
question.
beetles, can they regulate the chemical or the actual size or power of the ejection that they do, depending on the size of the enemy or size of whatever's threatening?
Yeah, so the chemicals that are ejected are uniform, and we've done lots of manipulations. Then that stays pretty much the same, but the quantity is quite variable, and the number of times they'll blast. And so it definitely depends on the strength of the stimulus, which you would imagine would be related to the predator and how it attacks. And they can also choose to shoot from only one side or the other. And so they can modulate that actually quite a bit. And then if you keep agitating them, they become desensitized. They realize that the chemical isn't going to work, and they go into other modes, either running away or playing dead, even though we found they still have some chemical left in them. So they kind of know when, okay, this is a waste of time. There's no reason for me to continue spraying. This isn't a strategy that's working. And it's probably expensive to make more. And so they go into other modes of action. So yeah, there is an interesting ability to modulate that.
So talking about expense and how long it takes to recuperate or to redo the chemical in their system, have you done any studies about that? How long it would take for them to produce the formic acid within them?
So we haven't done with the formic acid guys, but we have with the bombardiers, with the quinones and the hydrogen peroxide. And so you can get it. So like a lot of things for insects, it's temperature dependent, right? So it depends on what temperature we maintain them at. And if we keep them nice and warm and we feed them plenty, they can refill almost entirely in a week. And they'll have enough that they can spray within 2 days easily. Like even if you just keep at them and make them go completely depleted, they can spray again in less than 2 days under optimal conditions, and they can be full and replete within a week. If they're not healthy, if the temperature's lower, if you give, when I'm feeding them strange things, some of that, they don't eat quite as much, then that rate of accumulation decreases accordingly.
Very interesting presentation. Thanks.
Oh, thank you.
Hi, I find the abrasions on the back of the elytra to be, that's pretty neat. It's too bad that they won't do anything for you in the lab.
I know, it's very frustrating.
Yeah, it's almost as if, instead of being worn out from multiple males, it's also possible that it could be a single male, and if it's part of their courtship and female cryptic choice, and then you had that one that had a little tool in the same place, and so they may be doing something that's important to her choosing.
Yeah,
to use his sperm. But yeah, I guess keep trying with it. I don't know, keep fooling, making them happier or more comfortable.
I know. I need to put on the right kind of music or something. Yeah, so I definitely think you're onto some. So I have a feeling that it's like mate guarding. And so, occasionally you can find a mating, but you're like flipping a big rock, right? And you'll find them under there and they'll be clutched together, but they're not actually copulating. And I have a feeling that like copulation might occur when they're out wandering around and a male finds a female. He doesn't let her go, and they go under the habitat, and he's going to stay with her until she actually lays some eggs, but she's going to shake him off unless he keeps, giving her the right signal. And so that's like a mate guarding, and other males can't get to her as long as he's got a hold of her. I mean, this is, me making up a scenario right now, but I have a feeling it's something like that. And we haven't been able to replicate that kind of situation in the lab where they can go through this whole process and be holding on to each other long enough for that to occur.
So rather than being a product of some active rubbing, it could be just the fact that he's there for an extended period of time.
Perhaps, but it really looks like a rub. You can see like scratch, ones that are just scratches and ones that are like, more and more. And so it really appears like if I took a little tool and scratched the elytra of a pin specimen, that it would do the same thing. So I have a feeling there's something like a vibration or a rubbing that goes on, but we'll know till I actually get them to perform.
And I have another different question. In your drawing in the Brachinus, there was it was one gland on one side had one product and the other had another product that it mixed with. Is it really like that? So it's asymmetrical?
Oh no, it's probably just the way the diagram is drawn.
Oh, okay.
So both glands are symmetrical.
There's both on both sides.
Yeah.
Oh, okay.
Yeah. They're just mirror images of each other.
Okay. I was just wondering if it was that other way. That's pretty neat.
Yeah, no, nothing that everything is uniform, both sides.
Okay.
Yeah.
All right, good.
In the genus Stomus, the species had the name termite something in it. Does it have anything to do with termites?
So it's termitiformis. So I guess the describer thought that it had some general resemblance to a termite. I have to blur my eyes quite a bit to get that, but describers, it's in the eyes of the beholder, I guess.
And then in the mating, do you know what structure in the male or what part of the male does the rubbing? Do you have any idea?
Yeah, it's that last ventrite, the last visible sternite, because we can also see a rub mark on the male ventrite that corresponds exactly with where they make contact with the female. So when they're in copula, you know, that those two parts are in contact.
I got another question. What's the chances of identifying, I say Pteristicus in California? I've tried looking at some. They like I said, you said they look the same. They look the same to me. I mean, there's so much similarity.
Yeah, so there's two answers to that question. One is that there is no good revision for the Californian fauna, and the last time it was worked on by a guy named Thomas Casey, which I don't know if any of you know of Thomas Casey.
Yeah, you just took care of the. You
Yeah.
just convinced me that it doesn't work.
So Casey made it worse. Like, he just really made it worse. And so and in the north, when you get into Washington, you know, you have you know, work that was done by Hatch and Van Dyke and Lindroth. And so the northern ones you can do. Some of the California ones are very distinctive, but there's no key. And so I can identify a fair number of them, but that's because I've got types and I've been working on them. And so I feel really bad because at this point, I don't have the funding to do the full revision. It will happen. This is on my bucket list, like, but it's not there yet. And so particularly in Southern California, when you get into the Isabelli group down there, and some of the relatives of gliscans, which are on the islands, there's a lot of species, and there's also a lot of variation within species. And so you got the worst case scenario down there around LA.
And you said on top of that, there's a plenty without names, correct?
Absolutely. There's at least 2 dozen, 3 dozen. I just got another one last year in Big Sur. It's a mini Hyperpes. It's a very small one. Totally unlike everything else. So every time I get out there, I'm finding new things. So yeah, I mean, it's wonderful, but also I kind of would like it to stop.
I'm kind of familiar with llama,
Yeah.
but I haven't thought about this in a long time. Was it in a different genus?
Yeah, Holocephalus or something like that a long
okay.
time ago.
My recollection was I called it something else.
Yeah, but that's been
but I know that beetle,
synonymized for quite a while. Yeah. Yeah, it should be the state beetle, in my opinion. But anyway,
Oh, okay.
Could you talk about prey? Because you spoke a lot about the taxonomy, but not too much about what they feed on.
Yeah, so a lot of them seem to be generalist predator and scavengers. And so you'll see something like Pterosticus californicus, very widespread, doesn't mind disturbance, out in pastures, open habitat. They definitely will prey on other insects or spiders if they can get them. But also I've seen them feeding on seeds, on bay nuts that a squirrel has opened up and the meat is available. They're very much generalist. If you get something like Pterosticus llama, seems to be much more of a predator that seems to really go for ants and termites. And so you find them associated with the conifers throughout their range. So they're all up and down the Sierras and the Coast Range, in areas that have the kinds of pines that produce lots of termites and ants. And if you get into an area where it's just oak and bay, or if it's just redwood, the Pterosticus llama will drop out. And I think it's because they really prefer the termites and ants that are with those pines. So there's a big range of things of what we know. And then there's a whole lot that we don't know about, especially those subterranean things. I have a feeling that they are probably all good predators, and there's probably very specific things they're feeding on down there, but we don't know what it is yet.
Could you also talk a little bit about Calisoma?
So, Calisoma is we only get a few up here in the north, but you probably get the big black guys down there at your lights quite frequently. So in California, it's a relatively small diversity and density compared to, I lived in Tucson for a number of years. So Calisoma there would just cover the lights, and a lot of the Mexican species reach their northern limit in Arizona and also a little bit into Southern California. But we don't have so many here. But they're good predators, you know, often known as caterpillar hunters, but they do eat a variety of small invertebrates. In the Sierras, we have some that are very round and flightless. You might see some of these guys, very small species, and they kind of take the place of carabid, which is a big genus in the Old World, like over 300 species in the Old World. We have 13 in North America, 2 of which are introduced, but we have lots of Calisoma in North America, especially as you get into the Southwest and to Mexico. So I don't know if there's any specific questions you have about Calisoma, but.
Well, they could make a stink too. So.
Oh, absolutely.
Yeah.
Yeah. Isovaleric acid, methacrylic acid. It's a different form of chemistry, but they're potent, and they spray a lot. Yeah.
Okay, Blaine, do you want to take us out? You're on mute.
We've been trying to mute Blaine for years.
And I would enthusiastically welcome anybody who wants to take my place.
You did a fantastic job.
Oh, well, yeah. Well, thanks, Will. I greatly appreciate the talk, and we hope to someday see you again. And feel free to join us on our Zoom meetings. We'd be happy to include you in our mailings if you would like. And everybody else, it's been a pleasure. I will see what I can do about the December meeting and make sure that something happens with that. I don't remember what we have going in the next month. I'm sorry, I'm a little amiss in that, but I will try and get the next announcement out a little earlier than this one. This one went out a little late because I was traveling out of state. Does anybody have any last minute comments or questions?
Did anything pop up in the chat that I should have?
No, I don't see anything.
No.
Just
don't want to leave you wondering something about carabids before I go.
if anybody has a carabid question, we can refer it to Will.
And you're always welcome to send me images or specimens if there's something that turns up really interesting. So I don't mind that at all. Although I'm not going to turn around the hyperpes really fast if you send me a bunch of those hard Southern Californian species.
Well, I've got an occasional beetle or insect that I collect, and if ever I decide that I've got enough carabids that is worth sending to you, I might send them. The only drawback is they're going to be all in alcohol.
That's okay.
And
we can deal with it.
A lot of people don't like that, but everything I collect goes into alcohol.
That's
so
okay.
It's just my collecting technique. And although I use an aspirator a lot, and when I see a carabid, that's the last thing I want to aspirate.
Spicy, very spicy.
Yeah. That and a few species of ants are not pleasant. Okay, folks, unless anybody has anything else to say or do,
thanks, Kip. Fantastic.
Allow
looking forward
Kim
to your
shut us
review.
down.